Gostaria então de convidar
I would like to invite Dr. Rubens Lima, Professor Rayan, Dr. Gabriel Minero, Dr. Mariana Guimarães. She's a researcher of microorganism biogenetic for power plant species, invasive species, to be part of this roundtable. Welcome. Would you like to introduce yourself, say something before we start the questions? Good afternoon, everybody. I'd like to thank you so much for the invitation to be part of this roundtable with researchers that I mostly admire, presenting excellent content, and will contribute to a very productive discussion for us now. Good afternoon, everybody. I thank you so much, Mariana, Gabriela, especially that invited me to be here. It will be a pleasure. I have learned a lot from your presentations. I'm here for you for the additional comments. Thank you so much. Good afternoon, everybody. I'd like to thank you so much for the invitation, for watching the presentation. Clara, Paula, thank you so much. Rubens, if we have arranged, things would not have been so great because the content is converging. So it was very productive and we are going to learn a lot. Thank you so much and you are all welcome. Mariana, Dr. Gabriel, thank you so much. And Rubens and Clara, thank you. Welcome, Dr. Gabriel. You are here now with us. If you would like to introduce yourself, your initial considerations before the questions, please.
Hi, yeah, I was not sure. Something happened and the Zoom reconnected and now I can see myself in this group. So I was a bit nervous, but yeah, I am Gabriel Antonio Minero, as a scientist from Aarhus University, and I really like the format of this conference. I enjoyed the talks and looking forward to answer questions to my talk. Thank you.
Obrigada, foram muito, as palestras realmente foram muito boas, gente.
Thank you so much. All the presentations were excellent. Let's start with Dr. Rubens. There is a question for you, Dr. Rubens. Good afternoon, Rubens. We have difficulties. We cannot detect the difference in fluorescence, but we cannot make the difference between positive and negative tubes. Do you have any tip, the concentration, a cider? And Rayan has a question as well. Dr. Rubens, do you use nucleic acid before the reaction or just in the end of the reaction? Great questions. I will start with the final question and then the first. There are things that we can put in the beginning of the reaction, cyper gold, for example. It resists to thermal stress, and others no. Cyber green, we add it after the reaction. There is a trade-off. If you finish the amplification reaction, open the tube, there is a chance to become volatile in the room. If you're in a closed lab, in a short period of time, you have contamination in the environment. You cannot open mix of reactions in a room like that anymore. If possible, we prevent this practice. But if it's in the open air, that's not a problem. About the first question, this may happen for different reasons. In order to control, my suggestion is you try, I do not remember the name of the person, Julia Meirelles. Julia. My suggestion for you is to have another target so you make a difference if this could be specific for the target or not. Why should I say target? The prime can have a value of DEG that is favorable to the use of these primers, or a primer that is not so purified. Depending on the manufacturer, you can have different concentrations of salt and primers. They are not so useful in the amplification, but they are favored to the amplification that's not specific. And your result will be a cyber green positive, high fluorescence, but if this product you migrate to gel, you have a standard of band of lamp. So my 2 tips, try to see if it's specific to the target or if this amplification is purified or not. Thank you. Ryan, would you like to say something? Yes? The question, Rubens, thank you for your clarification. When we read the methodology of LAMP, they do not explain the details about the revelation, and then we are in doubt. We use cyber green, we use this, we use that. We do not see the methodology. Dilution is important. There are other factors that we do not see in the articles. And it seems obvious, but it's not. That's why I'm asking you, is it before or after? Because we see in the article, do not remember the species, they add cyber in the tip of the tube before the reaction. So that's why I'm asking, because the aerosols, as you said, not to contaminate, so you do not have to open the tube after the reaction. So we try to use coloring that is thermostable to prevent contamination in the environment. Very clarifying. Thank you so much. There are other strategies. Recently, they put cyber green in a pellet of agar in the tube, increase and heat, and after the heat, the agar melts, and the cyber green contacts the reaction. In my opinion, you have to include lots of variables, and then you bring more problems. When you simplify the situation. So let's think as simple as possible. Not everybody reveals in their publication what they do, their secrets. And just to clarify things, most of the outputs that are simple or cheaper, they are direct. The presentation, different intercalatings, I'll say, but ideally, if we could do is to design a probe, could be fluorescent probe, connected to the primers. As Gabriel mentioned in RCA, you have inside the reaction something that gives the specificity. That is LAMP as well in sleeps, but then you have to change the enzyme, you have to change the probe, and then you adjust the reaction. Ideally is to have a specific output. Sometimes you have to use a robust piece of equipment, but you have the guarantee on what you see. That's great. Next question is to Dr. Gabriel. This person is anonymous. I will read the question, yeah, Dr. Gabriel. Based on the experience that you have with these devices, do you believe there is a limitation in its application to detect a specific group of microorganisms from a multi-species biofilm?
Okay, if this question is related to rolling circle amplification, then I believe that one can use lots of different padlock probes to target different sequences in the cocktail of water, for example, with containing different organisms. So this is called multiplexing, and the only thing is you need to know what organisms are you detecting. If it's E. coli, you can target 16S RNA, but then you need to know the sequence of that particular 16S RNA of particular E. coli strain. Because even a small mismatch will affect the performance of a biosensor. But then if we go further, not only RCA, but another molecular amplification technique can be implemented in our biosensor. So I have a publication also using loop-mediated amplification, and my colleagues are working on recombinase polymerase amplification. And this centrifugal microfluidic system, for example, allows you to detect 16 species theoretically at the same time. So to wrap it up, you can, using the presented technology, for example, microfluidic system, detect 16 different species, but they must be of known origin, so that you can check the sequences and design your padlock probes or primers to detect them. Yeah.
Ok, muito obrigada.
Okay, thank you so much. We have one more question for you. From anonymous question as well. Dr. Gabriel.
Your very interesting talk. But at the end of the day, why did you decide for RCA technique?
Well, because we had the lamp presentation, it would not make sense to have two presentations on lamp. But yeah, I can compare with the loop-mediated amplification because I have worked with it as well. We experienced that these microfluidic chip systems leaking at temperatures required for lamp, say 65 or 68 degrees. The procedure we make them is that we use layers of PMMA cut using CO2 laser and glue it together using pressure-sensitive adhesive tape. Then we press it using 20 kilonewton pressure, and we hope that they are quite hermetic. And they are if we use mild temperatures. They also can handle 65 degrees, but the thing is, even one time the chip leaked, it created a big problem, just like Rubens and Rayan mentioned, right? Then what happens, you have to change laboratory because you cannot really clean, you try to clean tables and everything, but the false positive signal persists, so you change the laboratory to pipette your samples. And that's what people often don't really mention in their publications, how often they worked in the same university changing laboratories. And this biosensor that we developed was with the idea that we could implement it in quite emergency conditions where we don't have opportunity to walk between buildings. So rolling circle amplification in that sense operates at a lower temperature and can be integrated a bit easier on a plastic chip. It has a higher limit of detection though compared to lamp, but it makes it also more controllable because if the amplification is slower, then the occurrence of false positive signal is less probable. Basically, we can spill our liquids and contaminate the laboratory. You will not get the false positive signal because it's a linear amplification. But my colleague, he used different strategies to boost sensitivity by implementing, for example, several polymerases operating on the circle, or adding a step, say, stand displacement amplification, which is also working at 37 degrees. So rolling circle amplification can be also combined with the other downstream amplification steps, what makes this also attractive in diagnostics. So that's why we picked that one.
Eu achei muito interessante, principalmente.
It's very interesting to work with bacteria. A very important aspect that it happens very frequently is contamination. So when we can prevent it using this technique, it's something that makes our lives easier, and it's a favorable point to use this technique, in addition to the combination that we can use. We have one more question for you. Dr. Gabriel.
Your talk is very Indeed. Have you any thought for the environment species detection?
Can you please repeat the question?
Do you have any thoughts for environmental or species detection?
Okay, for environmental species, well, so I have worked for detecting pathogens, viruses, or bacteria, and I don't know what's meant under environmental species. But one time I was applying for a job to detect nucleic acids from ancient samples. So basically, they had a project, to collect soil from some rural area somewhere. And in that soil, they expected to find some ancient fossil or something, and they wanted to amplify that DNA material. But one of the problems with that was they warned me that the DNA of such sample would be very low quality and broken, and also there are lots of contaminants. So I would assume that when somebody asks me about environmental species, then we also have to think a lot about a real sample, maybe lots of crosstalk. Perhaps there's lots of nucleic acid material around, or some other contaminants. Then I would say, if I would have this task, then a proper cleaning, I would implement a proper cleaning of a sample. And if it's really one or few molecules of DNA there, then, my original talk was called capture and amplification. I would implement capture and up concentration of the sample. What you can do is you can use, for example, magnetic microbeads functionalized with short oligonucleotide that will capture in a specific way a sequence of interest from that environmental sample containing lots of different species. And then you capture it and you pull it using magnets, and you get your sample, let's say from a liter, you can collect those molecules and resuspend them in 100 microliter. It's a very big upscaling process. And then I would put it into the biosensors for rolling circle amplification. But all my previous concerns with whether this environmental species is of a known origin, or whether it's something that needs to be first sequenced, perhaps, then it must be taken into account. I hope it makes sense.
Okay, sim. Muito legal.
Yes, very nice, this answer. In fact, I have to ask you, all the participants in this roundtable, a person from our team is asking you, the person, would you like to hear your opinion? Molecular biology is more visual now in this moment. We are living in a bad moment, but what is the positive aspects for this area? Investments, the innovation in our area. What is your point of view? I believe that Mariana, would you like to say something? Mariana can say something and then we answer. I would like to take this question here exactly to talk about it. We are interested in detecting in the environment some species, not only with the interest in monitoring them, but also to trace some strategies that prevent the biofouling. And we use as a matrix water or sediment. We want to apply these techniques from this kind of matrix. I believe myself and Rayan, we have comments and doubts about it. How can we if we can have a good sensitivity in this technique, using this matrix, for example, water, we need to filter it to concentrate DNA and organisms in a membrane, and from this membrane, we can extract the DNA. So we need this previous step. Our interest is to use these techniques in the field, something more practical, easy to perform for people that do not have preparation in this technique. How can we extrapolate this approach for the field application, for the high power plant? And in fact, the question would be for both of you, Rayan and Dr. Gabriel and Dr. Rubens, based on your experience with these techniques. Okay, about... can I answer now? Yes, you can. I do not know if there is a sequence. No, go ahead, Mr. Rubens. Clara's question. I'm going backwards. We are so pleased this pandemic has brought us good aspects as well of the pandemic. That was, we know what RTPCA is, and it was Greek, ancient Greek, not long ago. People know what LAMP is now, among other terminations. But what happened, as far as I see, was a boom on preprint. Preprints are articles not validated without a reference by the peers, and they are great if they know how to read. In Brazil, we do not have an educated population that is critic to read and understand, and they bring something as true in a paper that is never going to be published, is going to be declined. This was a positive aspect, maybe on lack of education, at least in Brazil. I work here, that's why I'm talking about it. And this context that we have, the pandemic context, has brought us. I have this point to bring up to discussion. It was good scientific dissemination. People understand the tools of detection for molecular biology, and you also generate lots of fake news, misinterpretation based on preprints. About this point that Mariana has brought, now I will change to English. I know that you have the interpretation. I change to English. I talk directly to Gabriel. Maybe he comes after me.
Switch to the English, so now we can discuss directly. And I think that based on the point that was pointed by Mariana, I think that the main challenge of having these molecular biology tools in the field is the, how can I say, the pre-analytical steps. Even if you are working with human samples like blood, feces, urine, saliva, the main challenge is to have these samples enriched enough to be detected by all of these methods, so LAMP, RCA, RT, PCR, etc. So people say that this is very nice and field deployable, but when you have all your samples already at the end, but you have to prepare it before. And that's the main challenge for this, to apply or to democratize these DNA tests. For me, that's the main challenge for it. It's still for human, not only for environmental species, but also from human samples.
I totally agree with you, and thank you for turning to English, although the translator works perfectly. We also thought that these techniques, they are very powerful when it comes to synthetic molecules. The molecules often short parts of the genome that we order from a company and spike into buffer solutions. And then we use the commercial enzymes, which are also quite expensive. And we get these nice plots and claim those response plots and limits of detections. And then we fight, we kind of like that we could get a little bit lower limit of detection than somebody else. But I think that it's often a wrong way to pursue in research. That's why we turned, for example, to this loss sensitivity method, but we thought like to address certain challenges, and one of them, for example, is a presence of contaminants. Like you say, the sample first that we get will not be that clean molecule, it will be pretty much dirty. And then the lots of extraction steps will require people doing them. So this is where, for example, engineering comes, as I believe that interdisciplinary research requires molecular biologists working together with engineers who can develop maybe automated platforms or robots that can control these complicated reactions. And then I thought of, it would be really nice to have polymerases and enzymes that could handle these contaminants. That's why we used this phytoalbumin serum, but it was still like 5%, which you can calculate. That means that it's 5 microliter in 100 microliter. So it's still very small amount, but ideally we would like to spike, 90 microliter and some buffer so that we can get as much as possible of this sample, not just 5 microliter. And there's a company here in Denmark who develops enzymes, and it's called Novozymes. And I really wish if they would develop a polymerase that would not, that would be not susceptible to these contaminants. But there are so many different samples that we analyze. It's saliva, or it's, like you say, blood, urine, and all of them contain different contaminants. So in my opinion, the extraction is kind of a bottleneck to get clean sample. But I don't know how they manage to get these corona tests on a so short notice, because they get it from a mouth, and they must get it extracted and amplified, and the answer comes next day. So it is very good, but it's in centralized hospitals where they have lots of people who do it as their daily job. But I was just thinking, if it's about a poor country with outbreak of Ebola, for example, or dengue, forgotten dengue, right? Somewhere, in a developing world, then it must be a test that can be performed quickly and with a good, reliable answer, but not in a centralized hospital, but rather, in a field where we sort of have a tent and few people without a scientific background doing these reactions. Yeah. But I certainly agree with Ruben, and let's see what Rayan can say.
Muito obrigado pela palavra.
Thank you so much for your words. A little bit of Rubens said, this gain that we have here in Brazil, we were not so familiar with that. When you say PCR, people know what it is already. I go for a PCR. People say like this, it's good. This term is becoming popular. I think it was hidden, something so high, so difficult to reach, and this has contributed a lot and accelerated the detection process. And maybe, I do not know if I'm saying something or giving a question back to you. Within this that Dr. Gabriel said, when Mariana and I, when we think about LAMP, Clara as well, LAMP of the muscle, we always, we are our block is. What about the DNA extraction? That's our question back to you, because, the verification of the detection, the equipment, OmniLamp and others, they are more advanced now in this sense than the extraction aspect. And that's here, as Dr. Gabriel said, we have the collaboration aspect, engineering, how to put up a system, something portable, so we can have a good extraction of DNA in the field. Invasive species, in our case, 50 ml of water seems so little. How much DNA is there in 50 ml of water? And maybe, how can we detect the DNA of the golden mussel that sometimes is in larvae, 200 micrometers? How much DNA can I recover that? So talking about lab, if we get a larvae and extract, we can detect it for PCR. Quantitative PCR, we see that depend of the extraction kit, we can do it. But what about? I would like to hear from you. I have a question back to you, Rubens, as well, that have experience. And you raised this question, how do you think that we can detect this larva? Our big point of the invasive species of mussel would be this larva stage to anticipate this beginning of this invasive part. I do not know if Mari would like to say something exactly that. Our objective is to detect the very beginning of an invasion, very few maggots, larvae, very few. 5, 10 pieces, we have to detect in that sample of water. So it's a very small amount. Maybe after the extraction methodology, we can have an extra step to enrich that DNA, amplify the DNA, everything, and then we amplify of a specific target. I do not know, we have to adjust the best possibility. You mean to have a PCR of a PCR? So we try to reach a target. That's what you mean? No. First, amplify as you generate a number of DNA molecules bigger, and then you amplify the total molecule, not a specific. Enrich the DNA, maybe something that would be more costly, of course, more expensive. We have other limitations coming up now. Other challenges coming up.
Então, fazendo comentário sobre...
I have a comment about the final point, Mariana's point. It depends. If you want everything or nothing, the presence of the absence of the larvae, just part of the genome, is enough, but I understand your application. A pre-amplification, initial sequencing, but this would apply in a possible detection of a SNP, as Gabriel said. A very small amount, you cannot detect precisely that change. Let's imagine that is a species of mussel of a certain polymorphism that will generate higher spread, resistant to something. That's okay. A very small amount is possible to reveal these characteristics of that species. But if you want to know if there is presence or not, maybe the minimum amount to detect it would be enough for everything or nothing. Ryan, you know, this is a challenge not only for your area, detection of mussel in hydroelectric power plants, it's a challenge for coronavirus. The RNA simplers in the saliva.
Enfim, mas só o fato de você levar
When you take a Q-tip and transport the virus is enough to expose the DNA. Not DNA, RNA. Preheating is enough to expose. What I mean is, for coronavirus, LAMP is good because the pre-analytical phase is very simple. You get the saliva, heat up for safety of that sample. You have RNA enough to say that it's okay, that sample is contaminated, is everything or zero. But we know if you get the same sample the same way for extraction, especially using column. I know that we have good strategies for PCR in real time. Depending on the target, you need to use. But for lamps, you use the columns. It means in addition of the extraction, when you dilute in a smaller amount, tea or water, and this makes you to have a rich sample, and you have higher chance of positive sample. A lamp of saliva extracted is sensitive around 80%, comparable to PCR in real time of the same sample. So it's really sensitive just because of the extraction. If you preheat, you drop the sensitivity. We always discuss, you are right, this is the main bottleneck. Even if we advance, if we do not advance in the pre-analytical phase, we cannot advance together in order to place the solutions in the field. Rubens, I have a question. People who use lamp, do you have any news about the field use? How do you extract using it in the field? If it's in the field, they take to the lab later. Omnilampa works specifically to detect this new coronavirus. It's a diagnostic tool of COVID-19, but we do not have experience with other applications, although we have already developed some solutions already. But very soon we can answer that. The pre-analytical phase will be the limiting factor. If it's blood, depending on the target, you can centrifuge and get this overflowing and use as an input of the reaction. It really depends what you are looking for. For some application, leishmaniasis, you can have it. You can be sensitive enough for that. For Zika as well, blood, the virus is low. There is a point that the patient, you have a reasonable amount of virus in the blood, and you pre-process this sample to have the amount available. Every scenario, every target, every pathogen, we have different experiences. And even for the same pathogen, coronavirus, you see now, coronavirus is based on my experience, but you can apply to other. You see new variants of coronavirus now. It's very important to see different targets for the same pathogen applying. We use RT-PCR, this target, maybe it's not the best for lamp. And even during the experience with lamp, you have to select different targets to have a multiplex, thinking about increasing sensitivity. And this can involve the life cycle, depending on the phase, you have one target better than the other. It's the interdisciplinarity. So people in biology of the muscle, talking to the experts in molecular biology, and Gabriel as well, also engineering and physics. I would add physics to try to gather all this knowledge, finding a solution. Gabriel, do you have any experience with this extraction in the field? Anything that makes easier for us?
So the question was about extraction in the field.
Yes.
So no, well, first of all, I have never worked in the field myself, but I just imagine that such things happen in poor countries where people simply don't have, say, opportunity to get to the centralized hospital. If it takes, I don't know, a day to travel, right? So, but I would say I have ideas how extraction can be done, and one of these ideas I mentioned, if you use particles to capture the oligonucleotides. But I mean, there are publications to use this to up concentrate the material, but I personally don't have like experience, apart that we have one publication where we integrated this capture with further amplification and detection. And I can only say that if you capture thing on something, whether it's a particle or whether you immobilize it on a surface and then you say incubate it for 2 hours so that molecules get attached to the surface, and then you wash and you get these molecules nicely attached, then if you like to do amplification of those attached molecules, the system will suffer from the diffusion limitation, because the enzymes, they are suspended, they are not at the surface, and only few of them will be at the surface. So the efficiency of the methods will be compromised. So what needs to be done after you capture it, you need somehow to find a way to release it back, so that you capture something clean and then you release it to solution with all those polymerases and the reagents. Otherwise, the sensitivity will also suffer from the diffusion limitation of such a system. But also, since I'm speaking, I wanted to say to Ryan, I thought about what he was saying. Couldn't you, if you collect a sample of water and you would like to detect the golden mollusk, right? Or whatever environmental species that is presented in a very few copies, couldn't you sort of add some nutrients and let it replicate for a day and then try to analyze it? Would that work for you?
Gabriel, obrigado.
Gabriel, thank you so much for your question. You know, in fact, I joke that this lump project of the golden mussel, it's an embryo. We proposed this in our project, and we are still walking towards the amplification in the lab. We have not tested in the field yet. We do not see the larvae yet. We are still with the adults that we have DNA. We are standardizing first, but then we believe this euphoria of the scientists to want to see the product finished, analyzed. We think ahead, you know, placing questions ahead. That's a great tip, great suggestion. And this has to do with what Mariana said. Maybe initially this amount of DNA, because they are going to replicate more. This is excellent. The excellent suggestion. We can test it for sure. And the next step that we want to do in our studies is this: simulate, mimic a sample, environmental sample with water, with different amounts of larvae, so we can test the sensitivity of lumpy in the mussel. This would be our next step, yes. And I believe that this suggestion is wonderful. Maybe leave them overnight, going on this amplification, so we can have a positive result of initial DNA. It's worth testing. Let's take notes, Clara. Take notes. We are going to test it in our lab, this experiment. Yes. Thank you so much for the suggestion. Isn't it, Mariana? Yeah, that was perfect. Very important. Yes, welcome the suggestion. You are going to try it, and then we give you a feedback later. Now, I would like to ask Rubens a question, and Gabriel a question as well. Rayan and Mariana, of course, you are invited to be part of this question. I'd like to hear your experience, Gabriel, in the university, and Rubens in Fiocruz, about technological incentive. What about it in your area, in your countries, Rubens in Brazil?
You can go first, Gabriel.
Gabriel, you can go first.
Well, I'm not sure because it's a translated question. Could you help me? So it's about my experience, right? But experience with what? If you can repeat it one more time.
your experience with the investment in the technological field.
Okay.
Rubens, você pode... Consegue fazer um pouco melhor? Like innovation.
Okay, well, I will say quickly. Yeah, Denmark, I do research in Denmark, and they put a lot of effort into technology development. We have research institutes that try to bring forces together, and the Danish societies in general is pretty much a teamwork. So people work together from different disciplines. And there are lots of startups, as Denmark also tries to help young researchers to ground their companies. So one of those, it's called Blue Sense Diagnostics. They use actually this optomagnetic biosensors that I was mentioning, but they were pioneers. So they were working in the same group before I joined, and then they established this company. And now they have developed this sort of a product for detecting dengue virus, 4 serotypes plus Zika. So it's 5 pathogens at the same time. And I think they're quite successful, but there are also others who develop alternative ways to detect pathogens. And the fundamental research, like what I'm doing basically is a fundamental research. So I look into small things that might be of interest for me. It's not that popular, actually. It's very difficult to work in that direction. Much more popular here is when you can create a product, and people just try and fail. So this is very much appreciated. Nobody is perfect, and it's good to be in this kind of safe environment, that you have an idea, you get the funding for it, then you see that it's sort of working, and then you make a startup, and then one day you get a product. And if not, nobody is pursuing you to get you into prison because you promised and then you couldn't deliver. So speaking about my experiences, I think I really enjoy working here, and we have also lots of projects between university and the industry, because industry has like I said, they don't have enough resources to support, say, interesting but not very, say, applied projects. But then they can always work in tandem with university. So there is a sort of industrial PhD students or postdocs who work in the university, but they work for the purpose of a company. And I also think that this is something, the way to go, because this strengthening of collaboration between university and industry is something I experienced in Denmark. I was in Germany before, and it wasn't the case as to my experience. And I really can suggest it to everybody, right? Go for it, like do something that is relevant for in future for a product to solve a problem. But the world of industry is hard. They don't often they don't really support wild ideas or something that really might be like a thriving young researcher. But then there is opportunity to work in that direction in the university, while being sort of guided both by academic and by the company. So in addition to interdisciplinarity, like I said, engineers, physicists, and chemists, I also think that the future is in collaboration between academics and industry, as in my opinion, then the product will be like the best. I hope I understood the question, but yeah.
Sim, é exatamente isso. Gostei muito desse ponto de sempre ser uma...
Yes, exactly. I like this idea of a competition. It's the industry is not collaborative as the universities are. Really? Rubens, what do you think? My point of view, on top of Gabriel's question, I agree with him. Initially, I'm happy because Brazil can compete. At least we have brains over here. Brazil is a poor country, but we do not have a good management. We produce soybeans we are going to sell to China and the USA. So within this perspective, I believe that's good. Answer this in the scientific way. The outbreak of Zika virus in 2015, we answered very fast. Now, in this pandemic, Brazil could answer very fast in different aspects, the scientific point of view, but the funding, the way we can get this funding is not different from anywhere in the world. There are some specialties, but all around the world, they invest in the academia. The public sector invests. Germany, the US, who pays for it? Public sector does. So the communist that has an iPhone, that's great, because the technology, initial technology to build up an iPhone, they are here from the academy. I named some people, but I'm not going to talk about it now. Just to have an idea in this discussion, we cannot get away from the public funding in the academy to generate knowledge, but there is a limit. We cannot go to transform this into products without the industry help. And how can we do it? Unfortunately, in Brazil, we have a big gap between knowledge, what we know, and what is available to the population. So recently, I was discussing with people in a startup, old startup here in Brazil, one of the first in biotechnology, and they say, we are 15 years behind the knowledge. What we see today in the scientific articles, we are 15 years behind to apply this in a product. Other areas too. You see this boom on biotechs now during the pandemic, because we want to transform solutions, knowledge. We are talking about lab here. Before the pandemic, what was the tool of lab to detect disease? Very few. So we have this good aspect going on. For the other hand, we have a big challenge based on education in Brazil. The researcher needs a company. It's so sad. We want to invest, and we have this mindset of donating ourselves, work more than what we are acknowledged. Last year, I was out of this bridge with 2 colleagues, partners, in a company, a startup company. In the initial phase, the pandemic accelerated this process. Not me, we have other people in this situation. Every day, we overcome a barrier. The regulatory is awful. It pulls us back. Your institution is not acknowledged, it's not mature to understand that it's not bad, it's good. And I'm not naming anybody here. This is general, all the universities in general. We have a legal framework that is updated. People, the regulators, they are not mature enough to understand that the researcher can also work in the end, and it's not bad to receive for that. It's the opposite. We are simply trying to recompose what we have been doing for years and years, because we have been working for much less than what we should. That is a discrepancy. I'm just illustrating that is a discrepancy in Brazil compared to the rest of the world. The US and Canada, I lived in Canada, I see the biotechs there, and people finish their doctor thesis and go to biotechs. I start an economy course on a biotech. And then it's nice to see, for them it's natural. They are thinking as soon as they finish their doctor thesis, start up their own company, they are graduated for that. So we are so behind this level of education. Unfortunately, we lose brains for some spots, Silicon Valley, Boston, biotechs. So that's bad, that's bad. Brazil is so behind. And it's nice to see that we have brains to compete if we consider the academic knowledge to answer these questions that we have during this outbreak, Zika or coronavirus. That's my vision of the reality that we experience. About my work, my career, I came from Paraíba. I started a laboratory when I had leishmania culture without, I know exactly what is the restriction of resources for research, because I have worked in the Northeast region of Brazil. I came to Minas Gerais, the reality is different. 3 years in Canada, that is a different world. But something that never changes in this trajectory is to understand what makes things work are the brains, the minds. I talk to my students and they say, no, I'm not ready because they are so intelligent. Then a few months, they are teaching people from other countries there, you know? Something nice that the pandemic has brought. What we are doing, this internationalization. So we get closer to people and then we can see that a Brazilian person is creative. Since in the academic phase can contribute with some transformation. I see this during my career. It's not about budget. Budget is more than that. That person that is unsettled and wants to transform, there's no need for money. They need guidance, and then he's going to be in a hotspot in the world transforming the reality. Yes, it's true. The same as Rayan said a while ago. And I think that Julia, a collaborator, she went for her master's degree abroad and she saw the Brazilian researchers, they are welcome there. We believe it's because the difficulties we face here in Brazil with limited budget, in a bad manner, but it adds the way we solve problems. So the Brazilian minds are valued outside Brazil. Here in Brazil, the scenario is different. In general, we see this for music, Brazilian people in NASA, not only science, you know, in different areas. They work hard and they are very creative. Yes, they are. So keeping on this topic, there is a question. Dr. Rubens, you mentioned in some of your answers tropical diseases, they are neglected somehow. During the pandemic, do you believe that it will be simpler to have international partnerships for researching the tropical diseases? The natural and artificial barriers for these diseases are not so deficient. Professor Rubens and Gabriel, if you would like to say something about the tropical diseases. Yes, our experience, tropical, they are neglected. Tropical, this term is old-fashioned because of the barriers, global heating. Some of them, they are not tropical, and they are also different name. Neglected, when we say they are neglected, WHO, they say neglected, considering the attention. That does not mean they are neglected when we study them. Brazil is one of the countries that contributes a lot with the knowledge in leishmaniasis. So we are among the top 3 countries that produce knowledge for these pathologies. This does not mean that we are near a solution. We are not. As I answered previously, they are neglected not because the pharmaceutical industry does not want to find a solution. Of course, they want to find a solution, investing little and having more profit. So they are not going to have with neglected diseases because they are public sectors. It's bad to sell to the public sector, but the sales are big and they have big discounts. And you need to invest in P&D, research and development. One of the crème de la crème of the biotechs invest in cancer immunotherapy. This is very expensive, but you transform into medicine, and the person will take it and will spend thousands of dollars. It's a lot of investment to see a return of investment. For leishmaniasis that we work, this will only happen long term. So neglected, that's the sense, neglected of attention, neglected of this development of solutions. The solution that we have currently in Brazil to treat leishmaniasis was used first in 1947 and is still on the market. It's toxic, the parasites are resistant, we have lots of problems. And with the pandemic, for example, the diagnosis of leishmaniasis in the public sector was interrupted. The disease not exist anymore? No, this is just one example of many other diseases that we see in the media. Primary care is over. The disease is there. The priority now is COVID-19, but the disease exists, infecting, killing, generating morbidity. We are facing a dengue epidemic. It's not over. We are still facing it. Cardiovascular diseases, they kill people. People do not go to the hospital because they are fear of infecting with coronavirus. So this negligence will keep on like this. And the pandemic, one more time, is here opening our eyes to open the wound that we have these aspects. So these are challenges that Brazil will have to face many, many years facing because of bad management of the health system. Yes, one more time, I have answered most of your question, haven't I? I named the global heating aspect. Yes, it was great. I have a question during this discussion, and I believe this one is for Dr. Gabriel, asking if we should involve engineers since the beginning of the protocols, especially for the extraction process in the field, things like this. Sometimes some engineers can facilitate for the geologists, the biologists, and researchers.
Yeah, it's a very good question, and I think that I would say molecular biologists who design essays, they are often a little bit messy in the sense that they don't think too much about how it should be later applied in this kind of lab-on-a-chip systems. Then if this is not taken into account from the beginning, then by the time this essay needs to be implemented on a chip, some certain mismatches can happen, that maybe the volumes too big for such system to handle, or there's this issue called wettability, right? So say contact angle of what water forms on the surface. And one idea that we had was to have interconnected chambers so that we can sort of drag the extracted molecule from one chamber to another and then further to different chambers. And then the liquids were placed into these liquids and controlled using sort of phase guides. It's called some kind of, I don't know how it's called, but special modification of the surface so that liquid doesn't intermix between chambers. But when the assay was implemented on a chip, we found out that these enzymes, they need to have, you know, like 20 surfactants that needs to be present in order for the molecules not to aggregate. And this twin 20 has changed the contact angle of the liquid, and the liquid started intermixing. So what was tested on a simple colored liquids, that the liquids were perfectly connected yet not intermixed, was not happening once the real solutions containing enzymes were implemented. Yes, but thanks God, this all was quickly tested so that we didn't develop a lab-on-a-chip system that wouldn't be of any use. And yes, I think that these engineers and physicists must be involved in the very beginning, because if you propose, for example, or let's do PCR, then the engineer will tell you, but for PCR, we need to have a special materials that will not melt at 98 degrees. And also the liquids will expand perhaps at very high temperature, so that complexities must be taken. So let's go for some isothermal amplification. And that's how the thinking process can actually go and create a better, more robust solution. So I'd say yes, they should be involved in the beginning.
Realmente podia facilitar muito.
Really, it would make our lives much easier in this aspect of changing sites and sometimes create a recipient, a vessel for the PCR, prevent contamination. And also, there is one more question for you here, Gabriel. Anonymous?
I liked very much your presentation. Could you elaborate your techniques to detect biofilms presented in the environment, such as samples taken from industrial pipes?
In fact, I don't know, but you probably can see my background, right? It's my object of studies currently is a biofilm visualized in a confocal microscope. And you can see there are red cells. The red cells are wrapped in extracellular matrix that is green or blue, right? Green or blue is actually extracellular DNA that can be easily stained using some special fluorophores. So there is a lot of extracellular DNA in the biofilm. And my colleague is working on a project of detecting biofilms from the pipes. But what he does, he's only interested in detecting biofilm mass. So nothing like what particular pathogen is causing a trouble, because he showed me picture from the pipe and it looks so disgusting. There are lots of different pathogens and they form communities, and there will be lots of polymers and lots of things. And then if you would like to detect a particular species, you might ask a question like, first of all, the DNA is already outside. So the job is a little bit simplified because there's lots of DNA outside of the cells. But you basically collect this biofilm, scrub it from the pipes, and run the assay, and you will be able to detect. You might also consider to destroy the cells. If then you will also get genomic DNA. You can also separate sort of using mild surfactant. You can separate and extract the extracellular DNA and analyze both extracellular DNA and cells. Why? Because it's more like for fundamental studies. For example, if you would like to prove that the biofilm contains microRNA of a certain species, and this microRNA causes trouble. So maybe you would like to find a solution to destabilize these biofilms, and if you figure out that it's made mainly of RNA, So maybe it will help you to find a solution how to clean the pipes, right? And so it depends also what is your goal, just why you would like to analyze the biofilm, like presence of a particular nucleic acid material. You might design your assay in a different way. But there are certainly complex things, these biofilms. And what we were talking about, extraction, here it comes particularly because from the pipes, it's a very messy thing attached to the surface. And then you are looking like for the needle in a pile of things, right? So extraction will be a very important step in this case.
Posso completar o que o...
If I may say something on top of Dr. Gabriel? Yes, go ahead, Mariana. I think that it depends also on the type of biofilm that you see in the pipe. This would be another characteristics of attention. Also, the interest, the focus, the goal. For example, if the interest is in knowing what is the organism that causes corrosion in the pipe, I think that maybe it's simpler because it's easier to remove that biofilm out of the pipe and use the technique using a target 16S that is specific for a certain group, for a certain gender. But now, if the interest is detecting some microorganism that is present in the mud, that is another kind of problem that happens in the pipes that Rayan presented in his lecture. This would be something more complex because it will be associated to sediment. Biofilm is involved in the sediment like a gel filled with sediments, and that is another challenge different from a biofilm that causes corrosion. We needed to find alternatives to eliminate the substrate first, and then we find the target, maybe an organism that is forming primarily the mud. Some microorganism target in that mud. I think it'd be better to apply for the case when you want to know what is the microorganism that cause corrosion. This could be applied in this sense, but for sludge and mud, it's more complex. The final answer, there is one more question in the chat. Gabriel, did you research other ways to translate the signal in the biosensors? In what point of the C-shirt, the optical model is the best to identify nucleic acid? And do you think to use this technology for other kinds of analysis like antibodies, bacteria, cells?
So this technique is purely based on the optical anisotropy of magnetic nanoparticles, and it can be indeed applied for different targets. For example, what you would like to detect, you need to think what can be used to bind this molecule. So if it's a protein, right, then you can design aptamer that binds this protein, and then you have the same thing. You actually can you can create aptamer that binds this protein, and then you can amplify the sequence of this aptamer, and you detect basically the outcome of this amplification, right? Or you can detect it directly without amplification. The amplification step is good because from one molecule of protein, you can get 1,000 molecules of aptamers amplified. If you would like to detect it directly without need for amplification, then think what this protein can bind. If it's an antibody, for example, then you can immobilize antigen on the magnetic nanoparticle, and you will get the same procedure. We'll mix the nanoparticles, modify it with the antigens, and in the presence of the antibody, you will get this coagulation, binding of clusters to particles together, forming a cluster, just like with rolling circle amplification, where the nanoparticles were binding to the cluster of DNA. In this case, nanoparticles will be binding to the antibody because they contain the antigen, and vice versa. If you would like to detect antigen, you modify nanoparticles with antibodies. So, and I mean the guy who, the researcher who developed rolling circle amplification, his name is Matt Nielsen, and he has implemented this rolling circle amplification for detecting also cells and other molecules. And then basically what he was doing, he was transducing signal from cells, proteins into DNA, and then this DNA was amplifying using rolling circle amplification. And what we are doing, we detect rolling circle amplification products using magnetic nanoparticles. So we found that this is the best way because you can amplify the signal by employing the nucleic acid probes. Mm-hmm. But if I can say, think about optomagnetic technique as using gold nanoparticles. It's a very similar approach. Gold nanoparticles are also functionalized using tyrolated DNA probes. And when they find their target, they form clusters, and change in optical absorption from blue to red happens. That can be detected even by eye. But the problem with gold nanoparticles is that, you know, tyrolated probes, they require certain care. You know, when you attach tyrolated DNA to gold, you have to have, you should avoid presence of oxygen in this sample. And this tyrolated DNA is very easy to, how to say, to detach, because in all these amplification reactions, we have a mercaptoethanol. It's also tyrolated molecule present in high concentration that competes for gold with the tyrolated DNA. That's why the use of tyrolated, I mean, the gold nanoparticles in this particular assay wouldn't be very efficient. And that's why magnetic nanoparticles are more attractive. And you can also use a magnet to handle them. For example, what we have done in another project, we could use magnetic field to attract nanoparticles together and thus boost kinetics. Otherwise, it takes actually longer time for the particles to find their complementary target. But in the magnetic field, they accelerate it, so everything takes place faster.
Muito interessante essa, eu gostei muito dessa última parte.
Very interesting. I like this final part of the magnetic field, and I think that we need that. We use more these techniques like magnetic fields, implementing the techniques that we already use, that we already have, and improve instead of getting back. What should we do now with this DNA? What we should do now with these primers and these particles? Thank you so much, Dr. Gabriel. It was really interesting. Now I have a question to Rubens from Érico Freitas. Hi, Dr. Rubens. The structures asymmetries that you showed us, like lumpy, they are confirmed like a technique like diffraction, or these are just theoretical models? Thank you so much. That's nice, Érico. Thank you so much for your questions. Good to see you here. Your presentation in the beginning of the week. Érico helped me with some techniques in my doctor thesis. Thank you, Érico. Of course, he goes to physics. He wants to characterize the structure in the cyclic phase, you know? Depending on the target, imagine that we have 6 different primers up to form that figure. That's why it's a loop technique, the name. That is a loop because the loops are there. And then you have the primer loop that can solve and generate these sequences that are asymmetric. If you take one set of primer for one target, amplify and get this product, put in a gel, you see a standard of bands. And this standard of bands is specific. I'm not meaning that we are getting a standard of band of Leishmanian compared to a band of COVID and see that this difference in the bands. No, it's different of the conventional PCR. On the other hand, you can have a different standard, even with different targets in the same pathogen, depending on the region that you amplify, that standard can change. But if I can use this standard for a detection that is direct, for some physical method, type of signal, digital signature, this information, I do not have knowledge, but what I can say is that there is a certain randomization up to they organizing the most frequent ones. We need to get together and see if this is possible or not. But it's curious, this kind of question. And the answer is I do not know. What I know that these standards are specific of that region of choice. Is there a technique, Rubens, do you know, to find out? I'm curious. Imagine, it's diffraction. Imagine that you separate and you pass these fragments, capillaries, very thin, and the digital PCR that you can separate almost in units, virtual units, and they have a droplet, and the liquid in oil emulsion, and in fact, you can design a positive for diffraction, detect these structures. But if you compare A and B, 2 targets, we have similar diffraction, similar sizes. And I believe that to have a genotype using a technique like this, okay, it's relevant. But just based on the standard, I do not know if we have a digital signature, we cannot decipher the base. We are having diffraction of that size. So any 200 pairs organized in that loop structure, in theory, this would reveal the same result. On the other hand, if we can mark somehow a SNP and alteration of single base and apply this technique for a genotype, I believe that it's more doable. We do not need necessarily selecting these different sizes. We can have the SNP in different bands because the amplification of that initial phase will generate different other phases. And if we can mark using a physical technique and identify that SNP, this would be worth investing. Very interesting. And to conclude, Professor Rayan, there are studies of this biofouling can have any relationship with ionic ligature between the Bisu and Caspia cordylophora. Yes, I saw this question. This question is interesting. Thank you for the question, Saulo. And I joke when I say that in CBI, we know the golden mussel in our group since 2010, and the end of the project, we have the Cordylophora castia is our embryo. It is the newcomer in CBI for not a long time. So when we research about these other invasive species, it's well known in the southern region of the country, groups that worked already with some morphological aspects of these species in the southern region of the country. We have some level of identification in Pará, as amazing as it seems. There is a report of this in Pará. So there is a gap in the mapping of Cordylophora. Unfortunately, we do not have registered in the literature the interaction of these two invasive species. We have a single article from 2009, Portela, showing this interaction of Cordylophora and golden mussel, suggesting a potentialization of biological invasion of these two organisms together in the power plant. There is no article showing this interaction of Bisu and the strands in the ionic level. I'm sure that there will be some connection, ionic ligation, because the molecules are adhesive, glycoproteins, lipids that are involved, and ions are going to be part as well of this composition. The only thing that they see in this article, they have a longitudinal study, 1 year. Initially, it seems that in that site, Cordylophora is dominant in the first space and the gold mussel comes later. In the spaces left behind by the Cordylophora, when they open space, or on top of Cordylophora. We do not have this level of detail yet. And there is a collaborator, Professor Miranda, that she's willing to have Cordylophora in her lab. So we try and start, unveil the mechanisms and mimic the occurrence of their both at the same time. Unfortunately, we do not have is very, very little. We are starting up our studies. We are in the very beginning, detecting and mapping the Cordylophora in the country. As we see, she believes that it's spread all over the country, and we do not know this species. We do not have the characteristic, so we cannot show the true location. First, because it is an animal that is similar to a plant. There are filaments. We think that is algae, maybe bisu and mussel, but it's an animal, independent animal. And the relationship, the ecological relationship that Cordylophora brings and the mussel are dual, because sometimes they are together, cohabitating, and we also see the Cordylophora feeds from mussel larvae. This was seen in quagga mussel in North America. They are predatory, they live together, so complex. Cordylophora is a black box, we do not know. We do not know the answers. It's an organism with lots of possibilities for study, creating technology, and maybe Rubens to have a lab for Cordylophora as well very soon. And that's it. I do not know if I have answered your question. Valuable question. Thank you, Professor Rayan. Well, in this panel, the priority was the question to Dr. Gabriel, Dr. Rubens. These are external providers. They are so busy, we know. and it's a great pleasure to have them here with us. So we are moving towards the final of this discussion. I'd like to ask Dr. Rubens and Dr. Gabriel if they would like to say or ask or having their final considerations before we conclude this session.
Yeah, maybe I can say a few things. I really liked there was this question about what is topical disease, and then I was thinking, is, for example, tuberculosis topical disease or not? And basically, I wanted to say that what I don't like in the world of academics maybe is that we kind of follow the hot targets. So I can tell you that in 2015, when I joined this university, I worked with the dengue serotype 2, is where I used this LAMP technique, and we made a publication, but then we moved on because apparently the boom about dengue and Zika came to a very noise, basically, although I believe that it is still an issue. But it's just people were expecting that it will be like COVID, that they could have a high impact papers because they are combating something spreading very fast, but at the end it's not. And I feel sad because this is still a very interesting target to, because there are so many serotypes, and then you need to also be able to be specific whether it's Zika or not. But working with tuberculosis, I also was asked questions like, why tuberculosis? It's not really an issue, but it is an issue in also in developed countries. I met people who were treated for tuberculosis, so it can hit a person, and especially a person with a compromised immune system. So when we speak about targets and pathogens, I think there shouldn't be anything like what is hot and what is not hot. And I truly believe that all pathogens and microorganisms deserve these techniques. I mean, deserve a publication and deserve to be recognized and deserve to be analyzed, because whether they're good or whether they can cause a trouble shouldn't be a matter, in my opinion.
Bem, inicialmente eu gostaria mais uma vez...
Initially, one more time, I'd like to thank for this opportunity of being here. This event, I commonly do not participate, but this was really interesting. I thank you so much for the invitation. I apologize for my delay. I am awful to manage my time. And I believe that we have young people. I consider myself a young person. We are all young person here, most of us. And if I may leave you a take-home message for those that are young, and we have this boom, take this opportunity, not only for biotech, bioengineering in a general sense, and this could be the example to our country, so we can wave and surf this wave and show that we can really bring solutions, solve problems. Although we are in the academia, now it's about time to get together, industries and companies, so we take this knowledge ahead as a solution for the society. In different moments, we talked about these topics because in a second pandemic, I have heard Nicolelis this week, after the pandemic, there is another crisis, and he used another term that happened in 18 in the US, in India, and these deaf people buried, they contaminate the soil, the water, the food, developing different bacteria that are resistant. And it's not over, and it will not be over soon. We have to be ready for these types of outbreaks. thinking about the pandemic and afterwards. We warn people, fortunately, some countries answered, Brazil can never learn from them, and the only thing that is for us is persist. And my take-home message is this, it's not simple, but you are doing what you like. I hope that with these role models, these examples, we can take home this message that in Brazil we can be competitive, and today is in the hands of young people to transform this country. Thank you, Dr. Rubens, Dr. Gabriel, for your wonderful considerations during this panel. It was so important to us. And if there is no more consideration, we conclude this panel today. Rayan, I would like to thank you so much for your presence. It was a rich learning process to have Dr. Rubens and Dr. Gabriel here. And within Dr. Gabriel's considerations, this preference for some diseases, neglecting others, this is complex because as we have a single patient with that disease, it needs to be studied and treated, and it's important that we have equalitarian treatment. We get back to science somehow, never neglecting anything or anybody, because if there is this study, we have people suffering from it. This consideration is really relevant and important, and it's totally within the institute that Rubens Fiocruz, that is directly involved with so many diseases, and we see the managers, they do not give the right importance because COVID is here, but there is dengue as well, Zika as well. They are all together in this moment. I believe this is important. And thank you so much because you are inspirations. You bring technology and you move us towards. We want to go for new tools, new technology to solve our problems. They are not diseases, but there is a disease in the environment because an invasive species causes problems, economic and environmental problems. It's a different angle, it's a different point of view. So I believe that we can overcome this learning and go for pathology and ecology as well. So I thank you so much for your presence. I have learned a lot, and thank you so much for your availability, for your time, for your presentation, to be here in Sebei, that I admire this team. Thank you so much. Rayan, Gabriel as well. We always see our side. We do not think about other people that do not live, that do not suffer, you know? When we research this, we see always what is close to us. So I'd like to thank one more time. Thank you so much for this opportunity of discussing and learning from you. Everything was wonderful, productive. Rayan concluded very well. I shared his opinion. You are role models, so we can leave our box in the academia and show we have potential and we can extrapolate to the world, all the possibilities that we have. Thank you so much for everything, and good afternoon, good evening for everybody. Thank you so much for this wonderful panel. So, now we conclude this table, the final considerations of this third day of Fórum. Gabriel, would you like to say something?
I just wanted to say thank you, both Ryan and Mariana, for the wonderful words, and I really enjoyed this panel. And I was wondering whether the previous symposium was also online or you met in person.
Pessoalmente.
In person.
Oh. It would be a great opportunity to meet you all next year. Hopefully, yeah.
Eu acho que a gente pode fazer muitas trocas, né? A gente
can exchange a lot of knowledge. We can discuss a lot about our research. It's important to keep this ongoing. This would be wonderful. It would be a great pleasure to have you here in Brazil, Dr. Gabriel, in Brazil, maybe our next forum is in person. It will be wonderful to have you. We're going to be welcome to know our country and go to Minas Gerais for a walk.
Always wanted to go to Brazil, but the pandemic is going on. But yeah, once it's over, hopefully it should be over soon. Yeah.
Vai ser muito produtivo para todo mundo.
It would be great. So, we are concluding this panel today. If you do not have anything else to say, and let the students know some messages. We conclude the third day today. Tomorrow, we start at 9 a.m. BRT. And one more time, I'd like to thank you so much, the presenters, for their participation, everybody that was part of this panel, all the students that watched this forum. Thank you so much. And see you tomorrow.